Hello, everyone. Good afternoon, good morning to everyone joining us today. Our webinar today, Optimization of Phytosterol Crystallization, Influence of Solvent Composition on Product Properties, is brought to you by Paula Översti, Senior Specialist at Stora Enso in Finland. But before I introduce the speaker of today, I would like to go together with you through a few technical information, a bit of about the webinar that is being recorded, and it will be made available to you tomorrow or in the coming days. This week you will receive an email where you can rewatch it, and also if you are interested into reading again the slides, the same, you will receive them from us. They will be available on our website. During the webinar, if you have any questions, please make sure you use the chat question function of this platform. We will take them at the end, and I will read them to Paula, to our presenter today, and we will address that question. Also afterwards, if after the webinar, if you remember, oh, I have a question on this, or also for us, for Technobiz, you can write to us at info@crystallizationsystems.com or directly to Paula. I think Paula will also share her contact details, or we can also share the question with Paula later on. It will, the webinar will take about 30 minutes, and we will take questions at the end, as mentioned. Now, shortly, before we start on the topic of the webinar, for those that do not know Technobiz, we are a Dutch company located in the north of the Netherlands. We are a world-leading technology provider for solid-state research, process development, process chemistry, and formulation, with 4 products on our portfolios. Actually, I have to say, the crystallization systems portfolio. And I think Paula today will refer to one of our products and a case studies around our product, Crystal 16, from the crystallization screening portfolio. But since 2024, we have a new product line, and that's reaction screening with the Reactol. You might have heard about the Reactol. In a few words, what it is a multiple reactor system that is able to, in an automated way, to sample from reactor while you are performing the reaction, quench and dilute it, and transport it directly into an HPLC vial, so you can have your data from LCMS or HPLC. Any type of work related to route scouting, reaction screening, optimization, kinetic profiling, you name it, are able to be done, performed with the Reactol. But I don't want to go into too much detail there. I also want to emphasize to you, and maybe more relevant to this webinar, the crystallization product portfolio with the 3 products, Crystal Breeder, Crystal 16, and Crystalline. I have seen some names present already in the audience, and I think we are already familiar. I'm actually sure that you are familiar with the Crystal 16 and the Crystalline instruments, our instruments. Remember, Crystal 16 is your instrument for solubility, metastable zone width determination, solid state screening. You will hear also from Paula today. Crystalline with a camera technology that works exactly like a microscope. Also, if you have a Raman spectrometer, you can connect it to this instrument, and helps you to develop or optimize your crystallization or formulation process in the lab. The Crystal Breeder works at very small scale. Is, as I like to name it, gadget type of instrument that enables you growing single crystals, performing vapor diffusion experiments, controlled evaporation, And even you can, of course, crystallization study, perform crystallization studies at very low scale. Technologies, as I mentioned, is a manufacturer of lab instruments, which enables you to measure, characterize, and control some of the parameters, properties in crystallization, but also give you more insight for reaction screening, reaction optimization. And this is about Technoviz. I don't want to bother you with too much information about us. If you want to know more, you know where to find us now. Now I have the pleasure to introduce you our speaker today, Paula Översti. She is a senior specialist at Stora Enso in Finland. Obtained her master's degree in chemical engineer from Aalto University in 2012. After her graduation, she joined the renewable materials company Stora Enso. She has worked extensively in the R&D on pulp mill improved projects and in the development of new products and processes involving cellulose and lignin materials. Paula's expertise includes process design and simulations, techno-economic assessments, and profitability analysis. In 2021, Paula began her industrial PhD project in collaboration with the university, Aalto University in Finland, focusing on the design of phytosterol crystallization process. And I guess also actually her presentation today is related to her PhD. The research supports the company's targets for developing an industrial feasible purification process for a crude phytosterol-rich stream derived from tall oil or by a byproduct of the pulping process. The key research areas in Paula's PhD work include investigating the impact of solvent composition on the properties of the produced phytosterol particles, assessing solvent recovery systems, evaluating the techno-economic feasibility and scalability of the overall process concept. Paula, I cannot thank you enough to make this and bring this topic to our viewers today. Thank you for taking the time, knowing that you have a small baby at home, and in between all this and still bringing the topic from your PhD and maybe also work in the background. So thank you once again, and I'm going to stop my sharing. And I'm making you a presenter now, and maybe you can try now to share your slides.
So yes, let's try to put the presentation on.
Yes.
So
yes, now is perfect and is in presentation mode. Thank you very much, Paula.
thank you. Is it now that you can hear me well and you see my camera and the presentation is also on?
Perfect. Perfect.
Excellent. Paula. Thank you so much, Carmen, for your kind introduction, and welcome to follow this webinar also on my behalf. So the topic today is phytosterols, and especially how solvent composition is impacting to the obtained product properties. I come from Stora Enso company, but today actually I'm talking about my industrial doctoral thesis research that I've been doing at Aalto University. So let's get started then. A couple of words first about the content of my talk. So in the beginning, I will briefly go through project background and motivation. So basically, why is Stora Enso company interested in phytosterols? After that, there is a short introduction about phytosterols, just that it is then easier to follow the upcoming discussion and the results. But the main talk is focusing on the influence of solvent composition on phytosterol product properties. And actually there I would like to raise up three different smaller items what I want to focus. One of those being water. And another theme is then solvent composition in general and process parameters. And the third topic is hydration. After that, there is a short summary, and I think then we have some time for Q&A session. So, Stora Enso company, it's a renewable materials company and focuses on producing bio-based recyclable materials with low carbon footprint and basically materials that can replace fossil-based materials. Focus areas overall are in general on packaging materials quite broadly, but then also on building solutions and biomaterials and innovations. Wood is the raw material that we use in our operations, and basically when we fractionate wood into its main components, those being cellulose, hemicellulose and lignin, we also get like valuable byproducts out. And those are like crude tall oil and turpentine, for instance. Currently, we are selling this crude tall oil stream out as a product. But of course, as a company, we want to constantly investigate, can we do more out of our valuable raw materials? And we have been investigating disruptive CTO fractionation technology. And this fractionation technology is actually different compared to the traditional CTO fractionation technology-based distillation. So our technology can provide valuable products out of CTO with high yield and high purity. So this is really a fascinating concept, I must admit. And this would naturally then also allow us to take one step further in the value chain and practically make more out of our materials. The valuable components in CTO what we have can be classified as fatty acids, rosin acids, neutral oil, and then phytosterols. And for instance, I'm now focusing fully on this phytosterol purification process. So basically, after this fractionation, we get out a so-called crude phytosterol-rich stream, where we still have impurities involved. And then at Aalto, I'm focusing on this final phytosterol purification process design. Phytosterols in general are known to lower blood cholesterol levels, and the markets for those are in food, pharma, and cosmetic industries. We are targeting to get the product to food markets. There we have quite strict product specifications for phytosterols, and that's because there is only like few processing steps before the product enters the consumer. And finally, I must also say that I'm not the only one working with phytosterols, but we have several partners to work with us and especially support us in the scale-up activities for all these product streams. Some words about my work at Aalto University. So as I mentioned, the design focus is to design a purification process for this crude phytosterol-rich feedstock. And you can see in the picture that it's yellowish, slightly sticky material, and we have roughly 20% impurities involved. We wanted to focus on cooling crystallization technology as a purification technology. And briefly about the key objectives of my work at Aalto. So first of all, one thing is to focus on the solvent system evaluations and how they are impacting the product properties. Another theme is then optimization of the process conditions during the cooling crystallization. And there the idea is to make sure that we have good processability and scale up for the material so that we don't have like fouling or agglomeration issues. And obviously really important aspect is also this techno-economical evaluation of the whole phytosterol production concept. I'm not talking about that topic today, but just to give you an example that we can practically get the product properties in the required specifications with many different solvent combinations. But obviously, if the used solvent or solvent system is like too exotic or too complex, then this solvent recovery process may be extremely challenging. And then obviously that will lead to poor techno-economy. So it's really important to kind of keep that aspect in mind from the beginning. Then a couple of words about phytosterols in general. So they are heterogeneous mixture of plant-based sterols and stanols. And in this picture, you can see different individual molecules, sterols and stanols. And actually, all of the different molecules can have slightly different properties, like melting point. And there are like more than 200 different individual sterols and stanols, but just showing here now the main ones. If we start the production from a softwood-based tall oil material, so basically this beta-phytosterol is the main sterol what we have in the material. But obviously, if we use like vegetable oil-based feedstocks for the phytosterol production, then it would be that there are stanols which are the kind of major sterols or stanols in the system. So what I want to say is that depending on the used feedstock, also the phytosterol properties can slightly vary. And the properties are then combination of these individual phytosterols and phytostanols. So that's why it's good to remember that this is really challenging and heterogeneous material what we are working with. Stanols in general are sterol analogs with saturated forms, so they don't have double bonds in the structure. And as I mentioned that we are targeting to get the product to food industry, there is very strict requirements for the final product. I'm not going through all the details what we have in the specifications, but just mention the main ones what comes to our work. So basically overall phytosterol purity should be above 99%, but then there is clear shares for sterols and stanols, individual sterols and stanols, what we need to meet. And especially when we use softwood-based material, so we need to be really cautious that we don't have a final product where we have too much stanols in the product specifications. Then another thing is that phytosterols have different hydrated forms, those being anhydrate, hemihydrate, and monohydrate. And we can actually separate these by powder XRD, so they have different pattern there, but then also with TGA or Carl Fischer analysis, for instance, because of the water content is different. Then some words about the research questions. So when we started to work with the material, phytosterol material in the laboratory, we quite easily observed that many organic solvents actually, what we use in the cooling crystallization, fail to meet the targeted specifications for the product. And basically we used commercial phytosterol as a starting material, and you can see on the table the shares of phytosterols and phytostanols. And when we did the crystallization with pure organic solvents, for instance, ethyl acetate, acetone, or methyl ethyl ketone, so we always ended up having a product high in stanols. Then when we did the crystallization with a solvent combination, where we had a small amount of water, then we could decrease the phytostanol share in the final product. And basically that combination was best to fit our targeted product specifications. And we wanted to, of course, understand better what is behind this behavior. Is it because of different solubilities of sterols and stanols, or that can there be some differences in the hydrate formation? In the previous slide, I mentioned that there is, like in the literature, reported different hydrated forms for phytosterols, but actually there is no information regarding phytostanols. So that's why that topic gained our attraction. Another thing what we observed, unfortunately, was that when we have water in the solvent system, we result in extensive fouling and inhomogeneous mixing during the crystallization. So on this picture behind you can see that, well, this is a bit exaggerated figure, but still that the behavior becomes really fluffy and and mixing of that kind of material or pumping further, for instance, when you do upscaling, it's practically impossible. And obviously, we wanted to understand what is behind this. And also, can we, by tuning the process conditions, somehow impact to these obtained particle properties? Like, can we impact to the shape or size of the particles? And again, how is this crystal nucleation and growth during the crystallization? So in the beginning, we started to do solubility experiments, and we used Crystal 16 device for that. And this is like excellent tool when you have a lot of different solvent systems you want to study, and also if there is a lot of different raw materials to be checked for the solubility behavior. And in general, there is really a little information regarding phytosterol solubilities in the literature. So naturally, we wanted to start screening the solubilities with this commercial phytosterol mixture where the phytosterol purity is 100%, so that we know how it behaves as a pure phytosterol material, so to say. Then especially to evaluate the differences between sterols and stanols. So we produce this phytostanol material from this phytosterol mixture by hydrogenation process, so that we can then get understanding about these differences in the behavior, and especially this phytostanol material, how it behaves, because there is practically no information in the literature for pure phytostanols. Then naturally, later on, or the ultimate target is to develop this crystallization process for our real crude feedstock material, where we had roughly 20% impurities involved. So that was also what we obviously needed to test for the solubility experiments. But I'm not discussing about those results in this presentation. Then in this figure, you see what kind of information we can get out of this Crystal 16. And I'm fully aware or know that you know a lot of this method, so I don't need to explain too much. But just to highlight that the solubility information is supporting when we do basis for the crystallization experiments. For instance, we can easily identify what could be the good solute concentration to start with. And for instance, when doing cooling crystallization, what could be the like studied temperature range there? And again, for techno-economics, so we can evaluate or estimate the yield obtained, and then also how much do we lose phytosterol material in the mother liquor because of the solubilities, and especially if we don't like recover this mother liquor phytosterols in the mother liquor back to process, so that will automatically give us then the losses. Then some results. So basically how phytosterols and phytostanols are behaving in different solvent systems. So in this slide, you can see ethyl acetate, acetone, and ethanol as solvent systems, and the same ones including a small amount of water. So what we can briefly summarize is that water clearly decreases the solubility with both materials. Solubility increases with increasing temperature. But then most interestingly, phytostanols have much lower solubility when we compare the solubility in phytosterols. And when we investigate this a bit further, on the left-hand side, we can still see the solubilities of sterols and stanols in ethyl acetate and ethyl acetate water systems. But on the right-hand side, we can see the comparison of phytostanol solubility to phytosterol solubility. And practically, when we have like pure ethyl acetate as a solvent system, the stanol solubility varies obviously slightly with temperature, but it's around 20 to 45% of the sterol solubility. When we have water in the solvent system, so ethyl acetate water, the stanol solubility in comparison to sterol solubility is like 60 to 70%. So the solubilities are much closer to each other. This was really interesting finding, and then obviously we wanted to study that if the same applies when we do the real crystallization experiment. So we did crystallization with commercial phytosterol as a starting material, and we basically could conclude that when the solvent system was pure ethyl acetate, we get a product high in stanols, unfortunately. But when the crystallization was done with water ethyl acetate solvent system, then we got really nice profile for our purposes. So basically we had a much higher amount of sterols and lower amount of stanols there. And yeah, we wanted to analyze also the Madolico composition just to make sure that the mass balances are right, and then also that the solubility is the reason behind the different differences in solubility, and explains the different product properties what we get. Then some words about solvent system impact on particle properties. So practically, yeah, as we now learned that ethyl acetate water, as an example, is good solvent system to enable us to produce the product in the targeted specifications. But unfortunately, we have high agglomeration tendency there. And you can see from these SCM images that it looks really challenging material to work further. So we started to think that can we kind of replace the water, for instance, alcohol, and how would that behavior then be? So when we had ethyl acetate methanol solvent system, we got actually really nice particles, and we can quite easily and clearly see the growth of the particles, growth direction there. But unfortunately, then we had some challenges with the product profile, and especially when we did the crystallization with this real crude feedstock material. So we were kind of on bar with the targeted product specifications, but it was not that safe as with this ethyl acetate water. Well, then we wanted to investigate that what if we combine all these components, so ethyl acetate, methanol, and water. Actually, there we got nice particles, roundish particles, and also the particle size was quite high, so that the processability further was really nice and smooth. Also, this product profile, it was in specifications, so the sterol-stanol ratio was according to what we were targeting. And then also we had acceptable processability, so no agglomeration or fouling issues. So then we basically decided to move further on with that combination. But interestingly, we also found that there is a so-called two-step crystallization mechanism when we do the cooling crystallization. And that was basically observed with FBRM sensor, what we use during the crystallization to follow how the particles are behaving during the crystallization. But also we could observe that phenomena visually, and then also with camera-based setup, which we have been investigating as an alternative method. And actually on this figure, right-hand side, so you can see both this FBRM sensor, actually there in the crystallization setup, but then also this camera setup, but is then like through the window where we can observe the differences or how the crystallization is proceeding. And sometimes the challenge with this FBRM sensor, and especially this material, is that, for instance, this first increase in counts, it's really small, and it's challenging to detect only if we follow this FBRM total counts. So on the worst case scenario, so it may lead us to make some wrong interpretation regarding the nucleation point. So that's why it would be interesting to understand also what is happening there, and yeah, really verify this phenomena. So this two-step crystallization, we actually surprisingly noticed that when we modify this methanol water ratio, we can either get this two-step crystallization visible, or then we can go with a kind of more like traditional one-step crystallization. And it really depends on the ratio between methanol and water used there, whether it takes place or not. So what does it have to do with the final product? So basically we obtained that in the final product we have a p-modal particle size distribution if this crystallization goes through this two-step crystallization. And on the other hand, if we only have like one-step crystallization, then we have a unimodal particle size distribution. So of course we wanted to understand what is behind this phenomena, and that can it be that there is certain individual phytosterols or phytostanols crystallizing out in the first step, which could then result in this kind of two-step behavior. But unfortunately, we did not see any differences there in the products or product profile in the products separated after the first step or second step. So our conclusion is that it's not related to differences in the solubility or like different individual sterols or stanols crystallizing out on that temperature. Then another thing what we started to think about, could this phenomena be related to different hydrate forms? So then we did two crystallizations, one where the end product was after this first step, so around 52 centigrade, as you can see from the picture on the below, and then one crystallization where we isolated the product after the second step, so at 49 centigrade. And you can see this FPRM sensor, how the counts were developing on these crystallization experiments on the left-hand side. We isolated the products and then compared the behavior in this powder XRD. And actually we observed that there are some differences between these two materials. So that led us to think about that can there be some certain hydrate form actually crystallizing out at that particular temperature. And on that time we had only this FPRM sensor as inline analytical tool, but obviously later on, for instance, this crystalline or other like additional inline analytical tools could definitely provide some more information what is really happening there. Then some words about the impact of process conditions on particle size. So basically rotation speed. So we had ethyl acetate as a solvent system, and now I'm just showing this 300 RPM and 100 RPM SEM pictures, how it differs. So basically on the laboratory scale, this 300 RPM seems to be a bit too much for this system that we have studied. So basically too high rotation speed is breaking the particles. And obviously this mixing overall is something that needs to be optimized according to the used solvent system, used concentration, and of course scale and impeller that you are using. And clearly important parameter when you go up in the scale with the process design. Just really shortly about cooling rate. So there basically with ethyl acetate solvent system and these ternary solvent systems. So we compared 3 different cooling rates: normal, slow, and stepwise. Normal cooling is now 10 centigrade per hour, which is quite normal when using at industrial scale cooling. This slow cooling, we just wanted to kind of check that do we get actually bigger particles, because now there is well enough time for nucleation and crystal growth, and also we don't that easily attract impurities to the formed crystals. And then this stepwise method, so practically otherwise we used 10 centigrade cooling, but close to this nucleation point, and for instance now with this ternary solvent system through this first and second step crystallization. So we used slower cooling so that there was enough time to really observe what is happening and kind of let good enough time for the system to run. And then, yeah, after this second step, crystallization had taken place, then we moved to this faster or normal cooling rate, being 10 centigrade per hour. So we could see that with this stepwise methodology, we get largest particles. And actually also from techno-economical point of view, it's definitely better in comparison to this slow cooling, because obviously the crystallization time is then also much faster in comparison to this slow cooling. Then the final topic, and actually our recent publication, is related to this hydrate formation. So phytosterols, they are known to form hydrates, and we have also now verified that with several methods. So first we had the phytosterol material dried fully at 110 centigrade and vacuum. So we can observe in this powder XRD pattern that it forms anhydrate, and also that the TGA shows that there is no water in the structure. Then we wanted to investigate how is the moisture absorption with this material. So the same material was let store or stay at ambient conditions. And actually after 10 days, we still have this anhydrate structure in the powder XRD, but we observed that TGA, we had a tiny amount of moisture already in the product. But then if the product was staying at ambient 25 days, we observed clear differences in this XRD pattern. And also the TGA verified that there is some moisture in the system. And once more, when we had the phytosterol material first dried to full dryness, and then let it stay at humid conditions, so it quite easily absorbs moisture, showing 3% moisture in TGA, and actually also in the DSC, we can see this additional endotherm before this melting endotherm, but we obtain roughly at 139 centigrade. So from this, we can conclude that hydration can be modified by drying and storing conditions. So phytostanols, do they also form hydrates? And now we need to remember that in the literature, there is practically no information regarding stanols, and neither how they look at the powder XRD. So we didn't really have any reference point there. But basically what we then did, so the same as with sterols. So first we dried this phytostanol material fully. And with TGA, we actually observed already then that there is a bit of moisture in the product. But again, when we stored the material at ambient conditions 10 days and 25 days, so we observed that the material starts to absorb moisture relatively fast and much faster compared to sterols. And especially on these areas, you can see some differences in the powder XRD. Again, how is the time impacting there? So practically the same stanol material, but we first fully dried, and we kind of could think that there is anhydrate structure. We found in the TGA that this material contained 0.2 weight% moisture. And the same material already within 1 hour contained 1.3 weight% moisture. So this clearly indicates that this, first of all, the behavior is very different when we compare the sterols, but also that this anhydrate structure starts to change to hemihydrate already within 1 hour. So this is really, really different compared to sterols. And yeah, final slide basically regarding the presentation itself. So phytostanols, we wanted to investigate that can we then do some structure determination based on this powder XRD data, because as discussed, there is no reference in the literature. So the data what we got from powder XRD was analyzed by indexing to find the unit cell. And after that, we did this Lebel method to fit the results to a kind of full pattern. And this indexing activity, what we first did, revealed that there is actually two different structures in this material. So then we use two-phase profile fit for that. And yeah, on this table, you can see these unit cell parameters what we then investigated and identified for this hemihydrate phase and anhydrate phase. So actually for this exercise, this XRD measurement was roughly 2 hours. So basically this is then supporting the findings what we discussed earlier, that two phases or phytostanols absorbs easily moisture. So we ended up having two phases in this system. But yeah, practically the product is forming monoclinic unit cell, and it's a combination of hemihydrate and anhydrate. And shortly about the presentation still, so yeah, practically solubility differences between sterols and stanols results in different product properties. And especially we are interested in this sterol-stanol ratio there. Water, we noticed that it has a major impact on the obtained product profile, but then also on these physical particle properties. So practically increases agglomeration and fouling tendency. And that makes the upscaling extremely challenging, unfortunately. But then we investigated these ternary solvent systems. Now, as an example, this ethyl acetate methanol water. So practically that results in good product properties in terms of sterol-stanol ratio, but then also like processability aspects. Two-step crystallization, what we observed, is something that we could actually control by varying this methanol water ratio. And we think that that may have something to do with the hydrate formation on that particular high temperature. But there, it would be really nice to have some like additional process inline analytics to study this phenomena further. Hydrate formation differences between sterols and stanols. So yeah, clearly stanols are absorbing moisture much easier compared to phytosterols. So there was clear differences on the behavior, although the structural differences are quite minor after all. And I want to thank my colleagues and the team who has been working with me with this topic. So from Stora Enso side, Jari and Staffan, and from Aalto University side, Ping, Pekka, Esko, Eva, and Marietta. And basically this content of my talk today can be found more thoroughly from these three publications that we have had during this research. And yeah, finally, I would like to thank Technobics for inviting me to present my research in this particular forum, and thank you, audience, to following my talk. Although it's really interesting day in the Winter Olympics, also ongoing at the same time. And yeah, please, if you have any further questions or would like to reach out later on via email, so I would be more than happy to continue the discussions around the topics.
Thank you very much, Paula. Really great presentation, and I learned a lot about phytosterols. I knew very little about them. I have a few questions for you just before we stop this webinar. You are able to separate the stanols from the sterols, right? You do that by chromatography, or how do you do or not?
Now actually, we did with hydrogenation, so that basically all the sterols we kind of switched to stanol form. Yeah. But that is not actually what we aim then in the full production, but we did that only for this like research purposes, just to investigate more thoroughly the differences between these two materials. But yeah, that's possible to do.
Yeah, I was just wondering, because if you always have to work with the crude, so you always have a mix of sterols and stanols, and then also for the solubility and also for crystallization, and so it can be a bit of a nightmare, to be sincere. And I don't know, does the composition in the crude from the sterol to the percentage of sterols versus stanols, does it vary in the crude, or it's more or less controlled, the percentage, the ratio?
It can vary definitely depending on the raw material used in the process. So for instance, in soft food-based tall oil, so typically, yeah, there is certain ranges where the compositions are for, in general, let's say sterols and stanols. But that's kind of the material whom we want to develop this process further. So kind of we need to, I think it's most important that we analyze the raw material good enough so that we know that how it behaves, and then we start developing first of all the crystallization process, solvent composition, to keep in mind the starting material composition, and then also the targeted product specifications. So as I said, we are now aiming to food industry, so there is certain ranges regarding sterols and stanols what we want to meet, and that comes actually from the UE specification set for sterols. But obviously it can vary depending what is the final application you produce your material to.
Yeah. And I also I like your SEM images. So if I understand it right, with water you get into agglomeration. Yes. So, but then you still need some water to be able to lower the amount of stanols, right?
Exactly. Yes, that's absolutely correct. So otherwise, this water organic solvent system could be the perfect one, but then we have really challenges in the processability of that material. And of course, now I only showed you this ethyl acetate water as an example. So depending on the solvent composition, this product that we get out can vary a lot. But now we only studied those systems more carefully.
Yeah.
And actually, perhaps quickly mention that now these the solvents that we used, those were like polar solvents. So we didn't use, for instance, non-polar solvents now at all. But of course, that would then probably bring some differences then in the product obtained.
It's exactly what I wanted to mention, to ask you actually. That actually would be interesting. And it looks like these sterols and stanols, they like to make hydrates, anhydrates, maybe even solvates and so.
Yeah, that's something that we actually also tried to investigate, but at least we didn't figure out, or we could not clearly identify whether there was solvates or not. At least so far we didn't recognize.
Thank you very much, Paula. I think we need to stop here. It's almost 4 o'clock. Thank you for your time. Thank you for bringing such a beautiful case study where you exemplify solubility, crystallization, purification by crystallization, and so, and even impact on particle properties, shape, and I really enjoyed it myself. I learned a lot. I think you have shared your email address, right?
Yes, it was in the slide deck. Yes, but
yes. So
I think you will then share the presentation.
Yes. So if anybody has any questions later on, they can also write to you directly, but also they can also ask us at info@crystallizationsystems.com. Or I see somebody's writing on the last minute how to prevent oiling out during cooling crystallization. I don't know if you can say anything about this.
Oiling out.
Did you see any oiling out?
With some solvent combinations, I think we had a bit that kind of phenomena observed, but then I think we can kind of adjust the concentration, and then also how do we do the cooling so that we don't kind of crash out the material, so that we have controlled cooling rate, and then also that I would say that the starting concentration is not too high. But obviously that's something that definitely needs to be kept in balance. But I don't think that we have no, in the beginning we saw some phenomena of that, but not really lately. So
I must be sincere. I have seen it also in the past, oiling out in crystallization. It mainly appears when you have a multi-component system, like you have with more sterols, stanols, and so you would expect to face this problem, but indeed very much related to the solvent system or the mixture and the ratio that you are using. Yeah, it's just a matter of screening enough and finding out what is best for the specific compound product or system that you are working with.
exactly, and I think also because now we have observed that when the nucleation starts, so it can be really rapid, like phenomena that suddenly it kind of just comes out, so that we need to very carefully control the nucleation when it starts, and then that we can somehow try to slow it down as much as possible if it feels that it comes out too quickly.
Yes, indeed. And defining well the metastable zone width, I think that's also very important.
Yes, absolutely. And perhaps what I forgot to mention, so obviously there is more information than in this publication. So unfortunately now I didn't have time to go through all the research what we have been doing, but I think definitely with this material, the solvent system and how to screen that and how to modify then this crystallization is the key, how to make it work nicely and avoid this oiling out phenomena.
Yes, indeed. Once again, thank you very much, Paula. It was a pleasure to have you today here with us and to present about crystallization and phytosterols to our audience today. Thank you all for joining today, and see you to our next webinar. Have a good day or good afternoon, everyone. Bye. Thank you, Paula.
Thank you. Bye-bye.